friends, this is Preeti and you're watching my Solution. In this video, I'll be saying the general bacteriology chapter, this one, that is morphology and physiology of bacteria, important questions from this chapter. That is morphology of bacteria is important for 10 mark question, RS2. It has been asked in RS2 and 3 also. They ask about the structure, function of this bacteria, and then the antigenicity, distribution, and demonstration of flagella. These are the sub questions asked under this 10 mark question. And bacterial cell wall, the structure and the function, they ask separately for the 5 mark question. And this structure of bacterial cell is most important. This has been asked many times as a sub question under 10 mark question. Gram positive cell wall is important for 3 mark question, and even gram negative cell wall is also important question for 5 mark and 3 mark also. The characteristics and the diagram of gram negative cell wall. Then the demonstration of capsule is important for 2 mark question. It has been asked in the RS2 and 3. And cell wall appendages, this is important for a long essay, that is 10 mark question, and also important for a 5 mark question, that capsule. The flagella is important for 5 mark question, and fibrae and pillae are important for 3 mark question. Bacterial spores are important the diagram is important for a 5 mark question. And bacterial growth is the most important question. They ask you to draw the graph. This is a diagram. Then log phase. Under this, the log phase is important for 3 mark question. These are the important questions from this chapter. I'll be doing one by one. Because it becomes a lengthy video, I'll do the bacterial growth curve in this video. Remaining I'll be discussing in other videos. Bacterial growth curve. The bacteria are incubated in a liquid culture media. We incubate the bacteria in a liquid culture media. Then incubated, its growth follows a definite course. It follows a definite course. First, initially there will be lag phase, and then log phase, and then stationary phase, and the decline phase. It follows a definite growth pattern. When the bacteria's count in the culture media is different at different intervals of time, then we plot with a relation with the time. If we plot the graph with the number of cells in the culture medium, and according to the time, after time passes, how many number of cells will be there. Then we obtain a graph. This is known as bacterial growth curve. It has 4 phases. First phase is a lag phase. Lag phase means there won't be any multiplication of the bacteria. This is the period between the inoculation and beginning of the multiplication. Here the bacterial multiplication will begin. The time between the inoculation, when you inoculate, till the beginning of the multiplication, this period is called as a lag period. After inoculation in the culture media, they do not start multiplying. They have to prepare for multiplication. No? They do not start preparing immediately. So they take some time for the formation of the enzymes and metabolism. So the bacteria, the size will be increased, but the number won't be increased. Increase in the size due to the accumulation of the enzymes and metabolites. So there will be accumulation of enzymes and metabolites. Bacteria reaches the maximum size at this lag phase. This is all about the lag phase. What you need to remember, this is the time between the inoculation and the bacteria beginning to multiply. That is the time is known as lag. That is the lag phase. It takes some time. No, there no multiplication will be there, but increase in the cell size will be there because of accumulation of the enzymes and metabolites. Bacteria will attain the maximum size at this phase. This is all you need to remember about the lag phase. And now if we come about the log phase. Log phase, where the bacteria divide exponentially, and then suddenly there is an increase in number because it reverses exponentially. Exponential means like 1 become 2, 2 become 4, and 4 become 8. The multiples of 2 to power n, it will be going on. So this is a log phase where the bacteria divide exponentially. So you get the growth curve is a straight line. You can see in this image that you get in log phase you got a straight line. This is the log phase from the beginning of the multiplication till here. This is log phase where the bacteria's number will be increasing. At this stage, the bacterium size is small because instead of growing, that is continuously keep on dividing. No? So all will be newer bacteria, that is a immature bacteria. So the size will be small. And biochemically active bacteria, this stage they are biochemically active because they are continuously dividing. No? So they are biochemically active. This thing for biochemical any biochemical reaction, this is the best stage to see the biochemical reaction. They are uniformly stained at this time by the gram stain. They are uniformly stained. This is the best time to perform the gram staining. And this is all about the log phase. Log phase, what you do know, the bacteria starts dividing, the division is exponentially, so you get the straight line in the graph, and the bacteria size is small because they are continuously dividing, and this is the best method, that's the best stage to test the biochemical reactions. And gram staining is better, like best time for to perform the gram staining is this phase. This is all about, you need to remember the log phase. Long phase is separately important for the 3-mark question. And stationary phase. Stationary, after log phase, there is a bacterial growth ceases, like the bacteria was dividing exponentially, but now the bacteria growth is almost ceased because of exhaustion of nutrients. Nutrients will be available in a little quantity. Now after the consumption of these nutrients, there will be like a stationary phase because of accumulation of the toxic products and autolytic enzymes. Autolytic enzymes means that these are the enzymes that breaks the cell itself. These are the autolytic enzymes and the toxic products. These are accumulated in the stationary phase. So the bacterial growth, nutrients get nutrients, and other requirements like metabolites all are exhausted. So in stationary phase there won't be any multiplication. The number of progeny cells are just enough to replace the, like some cells will be dying. No, after the bacteria also has lifespan. Some cells will be dying. The multiplication, how it is takes place is the number of cells that are formed will be equal to the replacement number. So number of cells will remain stationary. So number of cells will be at the same level. Same number of cells will be there at throughout this stage. So this is known as stationary stage. Stationary says why this upgrowth is there means here it includes both the dead cells and the living cells. So it won't be stationary because the dead will be increasing and the living, so total cannot be stationary. So few will be dividing, no, so total number will be more, but the dead is replaced by these are living. The viable count means living count. Living, there will be stationary because the dead that replaced by the living, equal number is replaced. The enough number will be there, and hence the number of viable cells in the stationary phase remains almost balanced between the dying cell and the newly formed cells, but total count keeps on raising. At this stage, bacteria becomes gram variable. So we are not testing doing gram staining in the stationary phase. We are doing the gram staining in the log phase. And the more storage granules are formed. Sporulation occurs at this time. This is an important question for the MCQ. So sporulation occurs at this stage, and the bacteria produces exotoxin and antibiotics and bacteriocins at this stage. Exotoxins, antibiotics and bacteriocins. And a decline phase. The last phase of this bacterial growth curve is the decline phase. Decline phase is where gradually they stop the dividing. The bacteria cells stop dividing and the cell death continues. So there will be exhaustion of the nutrients. Why this cell death why this bacteria stop dividing means there will be exhaustion of nutrients and accumulation of toxic products because of the cell death. There will be decline in the viable count. But not the total count means like from starting how many cells were dead and alive also included. No, so that there won't be decline in the total count. Involution form is seen in this decline phase. This is all about the bacterial growth curve. We'll see. We'll understand the diagram now. This is a bacterial growth curve. In the on the y-axis we have given a number of cells, that is both death and alive cells. This is the number of cells. This is the time. This is the viable cells, that is the number of cells which are alive, like bacterial cells which are alive. These are total count. It includes both dead and available cells. This is lag phase. Lag phase means where there is a bacterial size will be increased by accumulation of the enzymes and metabolites. There won't be anything. Lag phase, and then they start multiplying, growing, multiplying. Then the multiplication number will be equal to the death number. So there will be replacement, and then they suddenly start declining because no longer existence of nutrients and metabolites for the division. But the cells keeps on dying. So this they declines, but total number keeps on increasing. This is all about the bacterial growth curve, and the factors affecting bacterial growth curve will be the oxygen requirement for the bacteria. There are so many types of bacteria. Like obligate aerobes means they compulsory. Obligate means like compulsory they require oxygen. Only they grow in the presence of the oxygen. Example, Pseudomonas and Mycobacterium. You can remember any two examples. And facultative anaerobics. Facultative is not compulsory for them to become anaerobics. They also grow in an aerobic medium. Example, E. coli, you can remember one example, and microaerophilic bacteria. They can grow in the presence of only low oxygen tension. And then obligate anaerobics, they survive only in entirely like absence of oxygen only they survive. Clostridium teni, this is an example. And temperature also is a factor which on which the bacterial growth curve depends. Bacteria's growth depends. Some bacteria requires optimum temperature, that is human body temperature 37 degrees Celsius. However, it may ranges for different bacteria, different temperature will be the optimum. Psychosporophilus grows best at 20 degrees Celsius. Example, Pseudomonas. And mesophils grows within the temperature range of 25 to 40 degrees Celsius. They are most pathogenic bacteria. And thermophilus means they require more temperature for their optimum growth, that is 55 to meso means middle, psycho means lower temperature, and thermo means at higher temperature 55 degrees Celsius. Geobacillus, stereothermophilus. These are the other important factors will be carbon dioxide, pH, light, osmotic pressure, and all this. I'll summarize this in this a table. That is, bacteria divided in bacteria division won't be there in a lag phase, it will be stationary. In log phase there will be there the bacterial division is more than the death, so that it will be straight line. At the stationary phase there will be it will be there, but it will be equal to the number of replacement cells, so there is no raise in this curve. In decline phase there is a complete depletion of the division, so the bacterial cell death will be there, so there will be the graph will comes down. And in bacterial death won't be there here, only bacterial size will be in log phase, it won't be there. In stationary phase it will be there, it will be equal to the bacterial division. And in decline phase there will be bacterial death. The total count, it will be flat, because number won't be increasing, it won't be dividing, then it rises again, it will be raising, but the total count will be increasing. But last at final there will be depletion of the nutrients, so there will be fall in the total viable count. Viable means who are live viable means living bacterial cells, that will be flat initially, it won't be increasing, no, then it raises in log phase, then it will be flat. Why? Because viable cells are viable cells, those who are live, that are replaced by the dead cells, so that the number will be equal, so it will be flat, and then again it falls because of the viable cells will keep on decreasing because of the cell death, because of endotoxin produced by the dead cells. Special feature, accumulation of enzymes and metabolites takes place. There won't be metabolically that will be active, but there won't be any cell division. In log phase there will be cell division, uniformly stained, and they have smaller cells because they keep on dividing now, so they have smaller cells. A gram variable, this is a gram variable, this is this is the most important stage for studying a gram stain, whereas the gram variable, it will be there because of accumulation of exotoxins, antibiotics, and bacteriocins. These three accumulation causes the cell death. And procedure is in this last one is inoculation forms will be present in the decline phase. This is all about the bacterial growth curve from Apurva Shastri. Thank you.